All right, so here we're looking at this graph that's commonly used and used in Chapter 8 and beyond Chapter 8. And we start with our substrate and we have our products in a chemical reaction that is going to go forward in this direction and reverse in that direction. So on this x-axis we could go forward to completion or reverse, and at equilibrium we might have 95% or say 98% products and 2% reactants. And the amount based on the amount at completion at final equilibrium, we said before, is based on the drop. And how much is the drop? The drop in this case is just simply calculated by saying, well, we start at 9, you drop down to 3, so the delta G in this reaction is equal to, and we have to pay close attention to this sign, it's actually a negative 6 kilocalories per mole. And why negative? Because we're releasing energy. We have energy potentially in the system and we're lowering it. So the delta G is going to be a loss of 6 kilocalories per mole. So these are the kind of calories that we burn when we digest our meal. Energy is being released in the form of kinetic energy, and this is the measure of that kinetic energy. We're focused in here on a single chemical reaction, but we could in a sense, sort of zoom out and look at the larger energetic flow through a system. And then look at this comparison here. So we could have this chemical reaction with no enzyme, and the reaction would go slowly. We could speed up the rate by adding enzyme A. We could speed up the rate further by adding enzyme B. What's going to be the difference in the amount of energy released? None. So when we add the enzyme, we don't get any more energy out, we just get it out faster. This could be the same enzyme, and enzymes can be modified. So this enzyme with its active site can be modified. Sometimes enzyme form B would be very active, and then there might be what's called a non-competitive inhibitor bound that would cause this reaction to slow down. And the reason it would slow down is because this activation energy would be raised. The enzyme would not be as effective like that. And this activation energy barrier is really significant. Another term that's used is that at the peak, here the maximum energy, that's called the transition state. And we've got to get it over the hump, we've got to get it over the transition state. And we can draw pictures of what the substrate looks like at the transition state, sort of the point of maximum instability. And what can happen is that the substrate can go up the transition state and it might not quite get over it and it could tumble back. So we have sort of the substrate sloshing around. And why is it sloshing around? Because it's holding heat in the system. So it's holding heat in the system. It's vibrating up and down in this kind of thing. Kind of like, imagine like the water is, you're kind of like bouncing it and jostling it, and the water's jostling around in the bucket, and if it gets up over the top, it'll tumble down the hill like that. So this is a key concept here, paying attention to this idea that the enzyme is lowering the activation energy, the transition state is lowered, and this would be the fastest reaction rate, but still the same amount of energy is released. You don't get something for nothing. Okay, next. The book is using the same drawing, same ideas, and so we're going over the same stuff in the book. And here is, just to show you this is protein synthesis. So we started out thinking a little bit about burning glucose and the idea that energy is going to drop. Here's protein synthesis. Protein synthesis, see, remember, we looked a little bit about protein digestion. It was hydrolysis. This is protein synthesis where water is removed. This is called a dehydration, removal of water reaction. And DNA, RNA, proteins, polysaccharides are all synthesized through a dehydration reaction. This dehydration reaction is going to lead to the formation of the peptide bond. Who's going to be doing this? The ribosome. So this is actually the chemical reaction catalyzed by the ribosome, like that. You know, we're synthesizing a protein, putting amino acids together, forming covalent bonds to make them connect them. Here, just incidentally, are the side chains of the amino acids. So we've got distinct amino acids being chained together. There'll be a few hundred of them together like that. All right, so here's a key concept. This reaction is energy requiring. How are we going to get that done? And there's a lot of that happening, right? We've seen there are a lot of proteins being synthesized, a lot of other molecules need to get synthesized, and they require energy. So here's this concept. A chemist would call an energy-releasing reaction often an exothermic or heat-releasing. We talked about how the calories are a kind of can be measured as heat or calories or joules work. Here is the kind of reaction that we're talking about to synthesize molecules, so anabolic reactions, and they're going to be heat absorbing. There's a more common term that biologists would use, and this is an endergonic or work requiring reaction, and this is an exergonic or work capable reaction, because so much of biological chemistry involves not just loss of heat and heat transfer, but actually coupling these reactions in order to get some work done. And the way to think about that is that we got basically water flowing downhill, and what we want to do is we want to capture that energy, so we could capture it with like a water wheel, and then we could couple that with another water wheel in which, say, something like milk is being pumped uphill. And that's what's going to happen in metabolism. Is a big part of metabolism is the coupling of release of energy from the foods we eat and using it to build molecules we need. And so there's no place in your body that has this direct coupling going on, because we use energy intermediates. So we'll see that what happens is we're going to burn down the foods we eat and release those calories, and we're going to capture the calories, not let them just disappear as heat. We're going to capture them by synthesizing molecules like ATP. And then ATP is going to be available floating around in the cytoplasm to be used to help drive reactions like this. So in the synthesis of that protein in the dehydration reaction, it requires some ATP, or we won't talk about this too much because the book focuses so much on ATP. There's another energy intermittent GTP, and GTP can be made in very much the same story and used to drive protein synthesis. We saw that ATP is used to move organelles, right? So there are two big energy requiring processes in the cell. One is chemical synthesis, and the other is physical work. So when you lift something up off the ground, you're doing physical work, and there's a lot of that occurs that's ultimately cells changing shape and muscle contraction. There's also a lot of that physical work that gets done, like when the macrophage is crawling. That's physical work, and it requires it's an endergonic process. So we can see an overall flow, and here's one way to see that in a food chain. So lots and lots of chemical reactions in photosynthesis. Again, this is going to be photosynthesis is going to be chapter 10, not covered on our midterm. But what's driving what is this synthesis reaction, endergonic reaction in a plant is sunlight. So that's the ultimate energy source, is we're going to use that to drive the synthesis of sugars that the plant can use for its own energy source. And then another thing that can happen is a bunny can come along and eat the plant. When the bunny digests the plant food, energy is going to be released like that, and the bunny can capture some of it, can harvest or harness some of it to synthesize the molecules it needs. Its body is going to be synthesized by the energy released from the plant it ate. Notice that this drop is bigger than this rise, because this actually is going to occur spontaneously. And that's a big surprise and confusion to people, that this life emerges spontaneously even though it has to build lots of molecules. And the way that does that is the same way that you could let a lot of water flow downhill and pump some milk uphill. Right? You could design a machine to do that. And this would happen spontaneously. The water would flow downhill, and as it did, the milk would be lifted uphill. And that's exactly what's going on here. We're coupling these two chemically through metabolism. Through catabolism, we are coupling that through a series of chemical reactions to anabolism. The next thing is, what if you tried to pump more milk uphill than you let water flow downhill? So what if the milk was going to try to go up that high and the water would be this high? You know what would happen? It would just make that thing run in reverse if it were a chemical reaction. We've got a forward reaction, we've got a reverse reaction, and the direction of the reaction is just based on the total net delta G. And there's a very simple way to calculate that. We'll learn about that in the next few minutes. If the net delta G is negative, like here, the net delta G is still negative. More energy is being released than is required to build a bunny. And that makes this occur spontaneously, because negative delta G reactions occur spontaneously, just like water flowing down a hill. So look at this. We can add this to it. Sometimes, I don't know, this is kind of gross, but it could be more of a hawk, but I suppose a crow or raven eating a little baby bunny is kind of gross. But when that happens, the bunny is digested by the crow, and the crow can build its body. But there still has to be a negative delta G, so this predator can't build as much as it digests. It's got to eat several bunnies, yay, in order to build its body, like that. And this is the energy flowing through a food chain. So we've really zoomed out. We're not looking at individual chemical reactions, but we're looking at the overall chemical reactions that are occurring and a food chain. And a biochemist sees that in a food chain, sees this energy flow through the food chain like that. So we've zoomed out to look at it at that level. Our book shows this idea of coupling chemical reactions. So it says, here's a simple chemical reaction, and it has an overall delta G of plus 3.4, like that. So it's endergonic. And it says you can make that, you could couple that to an exergonic reaction, like this is this energy intermediate ATP as a substrate, and if we release ADP, we're going to release 7.3 kilocalories per mole. We can couple these two reactions, put them together like that, and get an overall delta G, because we're going to pump something uphill and let something flow downhill. And that's all the book is showing here. And the bottom line is right here. It's just in the math. That's all we have to know about this, is that we can couple endergonic reactions with exergonic, and we just do the math, and we get an overall net delta G of minus 3.9 kilocalories per mole. And so this is going to happen spontaneously. This synthesis reaction here, the endergonic reaction, is going to occur spontaneously. And we could just plug in anything else. We could plug in that protein synthesis reaction, same deal. We're going to use something to drive that reaction. And really, really often, life uses ATP to push endergonic reactions forward. And this is typically the number. You might almost want to memorize that, minus 7.3 kilocalories per mole, because that's how much ATP is released, energy is released. And it can use up to that amount. It can push that amount in order to make the reaction go, like that. You can think about this, right? We talked about the drop being this delta G being the final equilibrium. So if we want this reaction to go forward, we're going to have to let the energy drop some to go forward. If it doesn't drop, the reaction will just be going halfway and coming back. And final equilibrium will only have will be product and half will be substrate. But the key thing here is in a complicated figure and description in your book, there is a bottom line, and the bottom line is just do the addition, keep track of the sign, and if it's a negative, then this will be a forward reaction like that. Each step in this from chapter 9 happening inside the mitochondria, burning down glucose, so a really common reaction in our bodies happening inside the mitochondria. We'll learn about it in chapter 9. Each of these steps is energy releasing. Boom, 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 like that. And some of the steps release so much energy that it can actually couple that energy to the synthesis of ATP. So in chapter 9, we'll look in the mitochondria at the synthesis, at the production of ATP. And then the ATP leaves the mitochondria and can be used by the ribosome to synthesize proteins, can be used by the motors that grab the vesicle proteins, and it can be used by actin and myosin and muscle contraction. So anytime you move, the muscle contraction is going to require ATP. So we'll have a place of synthesis during catabolism, and then we'll have its use to either synthesize molecules and build molecules, which takes energy, or move stuff around, like that, boom, like that. And so the book talks about this, that often there is a pathway, several steps on a pathway like we just looked at. So in general, we have processes like that. We talked before about how the enzyme will have a substrate. This enzyme binds substrate A. This would bind B. And so they'll have a different active site. This will bind C. So we need 3 different enzymes to get this reaction pathway done. We just step back here for a second. There are 8 chemical reactions here. We need 8 different enzymes. We shouldn't memorize the name of the substrates, the names of the enzymes, which is in your book, or the structure of these molecules. So don't memorize. But do understand that in some steps ATP is synthesized, and we know that it's going to take plus 7.3 kilocalories per mole to synthesize ATP, because that's how much is released. So we got the issue of microscopic reversibility. To make ATP, we're going to require 7.3 calories per mole. When energy is released, we're going to have a negative delta G, 7.3 kilocalories per mole. So what can we say? We could say something on a test about this. And I can use these terms on a test even though you don't have to memorize them. So the test question could be, succinyl-CoA reacts to form succinate during the citric acid cycle. That reaction is coupled with the synthesis of ATP. What can we say about this reaction? And what we can say about this reaction is that the delta G of this reaction must be greater than minus 7.3 kilocalories per mole. So that's a key concept that we do want to understand in this figure, and that is that if we are going to require 7.3 kilocalories to make ATP, in order for this step to occur spontaneously, we need to have more energy released. So we're going to have a drop that's bigger than 7.3 kilocalories per mole in order to drive the synthesis of ATP. Look over here. Here's NADH, and we'll learn that when NADH is made, it gets broken down to NAD. So NADH is high energy, and it drops down to NAD+, and that's enough that can be used to couple to synthesize 3 ATP. So ATP requires 7.3 kilocalories per mole, and so to make this happen, synthesizing 3 would be 3 times 7.3, or 21.9 kilocalories per mole. What can we say about this energy drop? This energy drop must be greater than 21.9. because we can use this huge energy drop to synthesize ATP. Take a step back, what can we say about this step? Here, NADH is being made. Boom. So that must require a huge amount of energy to push NAD plus up that hill to NADH. So consequently, this reaction, the reaction of isocitrate to alpha-ketoglutarate, must release a tremendous amount of energy. Look what's also being released here, oxygen. What's happening is we're really breaking the molecule down in these steps. We're simplifying the molecule, breaking it down, ultimately to release parts of it as carbon dioxide. So the carbons are going to get burned down and release carbon dioxide. This oxygen that's released, oh, sorry, it's not oxygen, it's CO2. I can't see that very clearly. So here's a step where CO2 is released, and where we're going to exhale that. That's what we're exhaling when we burn this down. This is why this is called cellular respiration. And this is dealt with in Chapter 9, but we're talking about the energetics of it and the principles of energetics here in Chapter 8. So again, figure from Chapter 8, the idea of releasing energy and releasing it in multiple steps. Same amount of total energy is released, but we have more control over it if we're releasing it in multiple steps, and this is where we could couple that with some synthesis. We can look at this, and we will in our next little video, and that will be our last video from Chapter 8.